Hi, and welcome back. So I know we already talked about the general structure of DNA, but this is intended just to kind of give you a little bit of a recap. So this is over the structure of DNA, RNA, genetic material, right? So DNA, RNA, right? So stuff that carries information about how to grow, develop, or organize an organism. So the two main polymers we're worried about right now are going to be RNA and DNA. So these are polymers of nucleotides, right? So they're relatively long polymers for the most part. RNA being less than DNA, DNA conventionally being much, much, much longer. DNA is a long-term storage molecule, it's a lot harder to degrade. However, RNA, since it still has its oxygen group, is a little bit easier to degrade, and that's actually useful when you're kind of turning over RNA in the cell. It allows you to recycle it quickly. So a nucleotide is composed of a couple of components. Number one, remember we need a nitrogenous base. We also need our 5-carbon sugar, and we're going to need a phosphate groups. Okay, so the nucleotides are going to be joined together by what we call the sugar-phosphate backbone. Let's take a look. Here it is. This is the DNA double helix. We looked at this a little bit earlier on in the semester, and so I just kind of wanted to bring us back to it. So remember things like A's and T's always pair together, just like G's and C's always pair together, right? So that allows us to have this symmetrical width throughout the entire length of the molecule, right? Because of the purine-pyrimidine size discrepancy. Pyrimidines being one circle and purines being two, right? 2 rings put together. So it gives us a total across of 3 rings altogether. All right, so that also helps us encode information. So instead of it being binary, this is kind of technically a quaternary. The sugar-phosphate backbone is easy to think of as this little handrail that goes up the side, and here they're shown in 2 different colors. One's illustrated in blue and one's illustrated in gray. So those are it shows that this is a double-stranded molecule, right? So there's 2 strands and they're joined together at the center by these little hydrogen bonds between the A's and the C's. And the A's and the T's and the C's and the G's. Okay, that's our generalized DNA double helix model. So let's look a little more closely. So this is just kind of showing that hydrogen bond here. This is again also the ribbon model. This is what we call that. So let's look a little more closely. This is called a partial chemical structure, and it gives us a general idea of how the atoms are arranged. So again, let's identify our pieces, okay? So the first part I wanted to identify here, phosphate group, right? Sugar. And in this case, this is how we get that sugar-phosphate backbone. So you can tell this whole section here and this whole section here, they act like a stabilizing structure. That's why we call it a backbone, and it's segmented, similar to vertebrae, right? So that makes sense. Okay, now let's move in a little more closely. So the hydrogen bonds. So if you'll remember, for A's and T's, it's the only 2-letter word we have out of A, T, C, G. So that means we've got 2 bonds. And also remember we talked about G and C pairing, and so that leaves us with 1, 2, 3 bonds. This is something you're going to have to kind of remember. All right, so even if we were to take off all of the letters here, you would still be able to figure out what the sequence is based on the fact that you still know a few things, right? You know things like your purines are pure as gold, meaning that A and G are your purines, so 2 rings. And you also know that pie cut. That sounds delicious. Let's cut some pies, right? So C and T, C, are going to be your pyrimidines, your single rings. Okay, so we can go back over to this structure and we can say, okay, 1, 2, 3 bonds. So that's a GC pair. So the single ring is a C. So this will be a C. The double ring, right? So that's the G. Let's see, here's a 1, 2 pair, so that's an AT pair. Our single ring is a T. Our double ring is going to be an A. right? And so that's how we can go back through, right? Oh, look, there we did. Pretty good, pretty good. And figure out the sequence based on the bonding pairs and the knowledge of purines versus pyrimidines. This is the sugar phosphate backbone shown another way. Well, actually 2 different ways. And this is important because I just want you to have an idea that we're going to show them quite a few ways. Actually, many of your study materials, including your book, will show DNA represented in multiple ways. So here's a simplified version, and this is more similar to the partial chemical structure. It still has the phosphate group here, phosphate group, phosphate group. We still have nitrogenous bases, and we also have our sugar, right? This blue area here, right? So each one of those are represented just in different ways, something to keep an eye out on. Let's look more closely to this deoxyribonucleic acid. So sugar for deoxyribose means we're lacking in oxygen, right? That stabilizes this particular structure and makes it a long-term storage molecule. Here's an example of a thymine, right? But we could have A, G, C, or T. Okay, so those are the ones we have for DNA. Also, we have this highly reactive phosphate group on the corner. So here's our bone right here, sugar phosphate backbone. And then here is our coding area. So DNA and RNA polymers are made up of nucleotides. However, they're not all made of the same thing. DNA has A, C, T, and G, which we've been looking at. Here's what they look like close up. Remember we talked about purines, pure as gold, double rings, right? And then pyrimidines, pi cut, CT, right here, single rings, just a little bit more. Also, in the structures, I want you to keep an eye on this. Like, there's a lot of nitrogens, hence they're nitrogenous bases. It kind of harkens back to a time when scientists believed that the oceans were or had more dissolved nitrogen in them, and so it was more readily incorporated into these early ring structures. Still is conserved today. So here's the difference between DNA versus RNA. So we talked about A's, C's, T's, and G's. But now we have something else we're going to throw in for RNA. RNA has A, C, G, but instead of having a T, we have a U. That's known as uracil, right here. Okay, so that's one of the ways we can tell that we're dealing with an RNA molecule, right? That's your big clue. Here we go. Now here's an RNA single there's a single unit of RNA. So here we have an oxygen, right? So this is sugar ribose. It's not deoxyribose. The oxygen is available. It makes it a little less stable, a little bit easier to degrade. And we also have a uracil right here, another tip-off. So remember, we're looking for A, G, C, or U to show us we're dealing with RNA. All right, well, I hope that was helpful. And if you have any questions, please feel free to email me. All right. Have a great night.